ऑन करना है ना अब नीचे।
Okay, I think the convener of NSF Colloquia is here, so I will start. Oh, I'm the convener, sorry. Sorry. Okay. What then you are the, I thought you are the convener. You're the host. Sorry, you're the host. Okay. Okay, so I welcome one and all for this Wednesday colloquium, which is a special one because we have one of our own, um, thankfully agreeing to give a colloquium, Professor Maithreyi Narasimha. She is from the Department of Biological Sciences. Just before we proceed with the colloquium, which is my long time wish to hear her, I would just like to give a brief glimpse of her background. Um, so Maithreyi actually is here for almost now 18 years, and in the institute, she actually has a background in undergraduate degree in medicine from St. John's Medical College in Bangalore, and then she did her PhD on genomic imprinting in the mice with Azim Surani at the University of Cambridge. And after two postdoctoral stints at University of Cologne, Germany, and University of Cambridge in Nick Brown's lab, she came back to TIFR, where she has been looking at Drosophila morphogenesis. And this is something that has led to a lot of very interesting discovery from our lab. Um, in uh, uh, sort of unified way. When I heard Maithreyi for the first time, this was I joined in 2010, so this was a TIFR Wiseman meeting, and I had heard about mechanical responses of proteins being measured for the first time by Carlos Bustamante in at Berkeley with tweezers. And that was the time when people suddenly looked at mechanical properties of biomicromolecules. It turns out to be that is important. Biology had not realized, at least the contemporary biologists had not realized the importance of these. But Maithreyi's talk, when I heard for the first time, really told me that collective behavior of cells and tissues are governed by these mechanical sort of stresses in between cells, tensions in between the cell, and that has pretty much have very important influence in developmental biology. So, Maithreyi, this is my long-standing wish. Thank you very much for doing this, and I look forward to your colloquium.
Thank you. Yeah, thank you. Thank you, JD. And it's his birthday today, so I have a lot of pressure to performance pressure, performance pressure maybe. So yeah, thank you for inviting me to speak here. I've actually not spoken here since a few months after I joined this institute, and that makes me feel ancient already. So what I'm going to do today is try and give you a flavor for the sorts of things we're interested in the lab. And in particular, I'd like to tell you about how you shape and move living structures. So I just begin with a few definitions. Morphogenesis is a word I'll keep using throughout the talk, and it comes from Greek that literally means the creation of shape, morpho for shape and genesis for creation of. And it refers to all the processes by which organisms, organs, and tissues within them, as well as the cells that make up these tissues, acquire the shape or form that's critical to their function. You can think of form or shape as being the defining feature of different types of organisms. The dog has a shape that's different from the human. But what I'm actually going to talk about is the creation of shape in a developmental time scale. By that, I mean, how does an organism of a particular species, shown here as a human, acquire its final characteristic shape, right? That you can recognize as being human. And the problem here is that the beginnings of the shape are very simple in that it's a single-celled zygote that's formed from the fertilization of the egg and the sperm. And in fact, if you look at the first 25 days of human development, I mean, these little dots here really look like flecks or specks. But if you could look at them at a cellular scale, you will see that this single cell, through divisions, becomes a multicellular entity, a loose aggregate that then, I don't know if this thing is, anyway, becomes a loose aggregate in which cells then begin to make contacts with each other. You'll see after a while that you can distinguish more than one type of cell, the red one here that's on the inside and the yellow ones on the outside. And a little bit later, there'll be the formation of layers and cavities, that many layers, many cavities, and so on. This is just in the 23 days of early human development. So clearly, the things that we can see accompany this process are an increase in cell numbers, an increase in the number of types of cells, but also changes in the positions of cells that eventually contribute to this geometric complexity. And if
Sorry, he will give you a regular pointer. This is not working out.
And so what I want to say is that if you look at the early development of, this one here, right? Yeah. If you look at the early development of other organisms like the frog, it's not that different. You have cells, multiple cells, layers, cavities. And this is also true of the humble fruit fly, our favorite model organism, in which, although the egg might look elliptical rather than round in its early development, you still see that one cell becomes many, you form one layer, you isolate some cells out as being different, then you form multiple layers, and you form cavities. So typically, we think of morphogenesis as occurring through the regulation of cell numbers by cell division or cell death, regulation of cell types by a process we call differentiation, and the regulation of cell shape, cell size, and cell position that I'll collectively call morphodynamics. And it's become appreciated now, as JD said, that morphogenesis relies not just on molecules of the kind that regulate gene expression or the ones that mediate signaling or facilitate interactions between molecules, but also by physical principles, notably mechanics. And I'll spend a little time talking about how molecules inside cells can aid generation of forces. So we think of morphogenesis as requiring the interplay between molecules and mechanics. It also requires integration of information at a number of length scales, from molecular to cell and tissue length scales. And this in itself is not a new idea. And physical principles and mathematical principles were put forward as a basis for pattern formation or form generation long before genetic principles were attributed to it. And D'Arcy Wentworth Thompson wrote in 1917 that biological form can reflect physical and mathematical principles, and argued that a certain mathematical aspect of morphology is not just helpful, but actually essential to the proper study and comprehension of growth and form. I'm going to skip a lot of history, but just to say, Stuart Newman wrote a book called The Biological Physics of the Developing Embryo, in which he argued that the development or the shaping of diverse organisms is made up of stereotypic morphological motifs, and I'll come to that in a little while. But these morphological motifs include the ones that I just described, which is layers, cavities, aggregates, and so on. And he said that the way these way in which tissues sort of elongate or fold or segment, the way in which tissues go on to do all of these things are similar to processes that are generic to condensed, chemically excitable viscoelastic materials, right? And he argued that embryonic mechanisms that generate them are typically much more complex. And he was one of the people who sort of pioneered the thinking that the mobilization of physical organizational effects must be responsible for the complex patterns that you see during morphogenesis. So the idea that genes actually dictate the shape of organisms or the shapes of tissues within organisms was relatively new, and it had its foundations in the Nobel Prize-winning work of Yanni Nusslein-Volhard, with whom Mahendra did his postdoc, and Eric Wieshaus, along with Ed Lewis, who performed what we call mutagenesis, which is to take the genome of an animal and mutagenize it so that some base pairs of DNA are not what they should be. And then through that, looked for mutations that cause animals to have a change in their form compared to the normal form. And through this, they identified genes that regulate form and first established that genes are determinants of spatial organization in developing animals. This led people then to ask, if genes regulate shape, how could they be doing so? And in particular, people began to ask, what must genes be doing to cells in order that you can regulate the shape of a tissue? And this led to an emphasis on asking, what are cells actually doing? How do cells form sheets and cavities and layers? How do sheets elongate, contract, fold, and flatten? And again, as JD said, a number of new techniques that have allowed us to visualize, manipulate, and measure both molecules and their physical properties have led to the resurgence of what you might call the physical biology of morphogenesis, although this had its beginnings in the early 1960s, largely on cells growing in culture. Okay, so now if you,
This genes that you are saying that it controls the shape, is it the individual cell also has a certain shape, or the growth like cell divisions which are controlled in certain pattern? Is that?
Yeah, so I will sort of come to that. So I'll show you that every cell has a three-dimensional shape. This shape is not necessarily static, but can change over time. And within a tissue, even if you have genetically identical cells, you can have cells of different shapes, right? So that is part of what I referred in my title as morphological heterogeneity, in that in order for a tissue to be sculpted to a particular shape, all cells within that tissue cannot necessarily be of the same shape. So let's say you want to make an elliptical tissue. You have to do something different at the edges of the ellipse compared to the center. And I'll show you how that becomes important. So how can cells actually change their shapes? So if we think of the cell as the minimal living unit of an organism or a tissue, it becomes important to look at how you can alter the shape of a cell. And we think of one key mechanism, which is linking the membrane of the cell, which is what covers it on the outside, with the cytoskeleton, which is the sort of force-generating machinery inside the cell. So if you can link the membrane and the cytoskeleton, and the cytoskeleton generates forces, then you can deform the membrane. And by regulating when and where the cytoskeleton is linked to the membrane, you can generate a variety of shapes. So if you look at the membrane is a self-assembling phospholipid bilayer with lipid and protein mosaicism. This is something we learned in school. And its protein and lipid composition can influence its physical properties, including things like rigidity and curvature. The proteins can themselves be organized in a mosaic way. So they may be present in one part of the membrane, but not in another part. And this can lead to localized action. And a specific class of proteins on the membrane, which we call adhesion molecules, allow cells to make contact with each other or to a substrate on which a sheet can sit. So here in green are the outlines of two cells. And what you see here bridging across these two cells are one kind of adhesion molecule, a protein that enables the formation of contact between two cells, and a physical property that this contact achieves is to reduce surface tension. Another class of proteins, which you see here, allow cells to interact with a substrate. It's a bit like my foot on the ground, right? If I were holding my hands, somebody else who's also on the floor, then that would be through a cadherin-like adhesion. If I'm walking on the floor, I would use these proteins that allow the generation of traction forces against a substrate. Both these types of proteins have one thing in common, which is that they link through their insides, meaning the parts of the protein that's inside the cell. There's a part that juts out of the cell, as you can see here. The parts that jut into the cell link up to this cytoskeletal machinery. And by so doing, you enable a way by which you can link the membrane to the cytoskeleton. And there are many different ways in which you can link the membrane to the cytoskeleton. And if these blue and gray lines are the cytoskeleton, you can sometimes link them with only a subset of these other proteins that are shown here. And that diversity can alter the forces that the cytoskeleton can generate on the membrane. So the cytoskeleton itself is sort of essentially three systems of self-assembling polymers, and I'll focus on microtubules and actins. And their network organization depends on other proteins, interacting proteins. And their ability to generate force depends on polymerization against a barrier, for example, the membrane, or anchoring to the membrane, right? And this may or may not require the action of motor proteins, the kind that you've heard other people talk about. Myosin works with actin to facilitate contraction. It can also facilitate expansion depending on how the actin filaments are oriented relative to each other, and so on. So there are a number of things one can tweak to generate different types of contractile networks. And microtubules like actin can also generate force by polymerizing, or for that matter, depolymerizing against an obstacle, or being anchored to the membrane through a complex of proteins. So what we've been doing we've been doing in the lab is to use the power of the fruit fly, the same maggots that come and feed on ripe mangoes and banana, to try and understand how tissues are shaped and how tissues are positioned. We use the fruit fly because it's a multicellular organism. It's made up of many cells. It's genetically tractable. It has 100 years of genetics that were pioneered in very early on by Thomas Hunt Morgan and by people that followed him. It has a fast life cycle, good breeding, it's cheap. And over the years, people have shown that fundamental mechanisms that operate in, for example, shaping tissue, shaping cells, are conserved between flies and all other organisms. And so the other point I want to make is that the way in which the fly embryo develops, and I'll show you a movie about that in a little while, provides us with a diverse array of morphogenetic transformations and morphological motifs that are accessible not only to visualization by imaging, but also to perturbations that are not just genetic, but also biophysical. And so you can look at how this fly embryo develops. And this is a movie from Philip Keller at Janelia Farm. It doesn't start at the beginning, but at a time when you've already got a large number of cells. Each dot that you see here is a single cell. It's the nucleus of a single cell. In this part here, you're looking at the embryo from the top, or the back end, and this part you're looking at the embryo from the bottom or the front end. And when I play this movie, you will see how much movement, deformation there is that accompanies morphogenesis. So if you start watching here now, you'll see that this part of the embryo, which is really the axis of the embryo, moves from somewhere here all the way back to this end, right? This is the head end of the embryo. This is the tail end of the embryo. So something that was folded up into a U-shaped structure actually unfolded and made its way from the head end back to the tail end, right? What you'll also see is a little elliptical structure here that becomes smaller and smaller with time. And at the end, you'll see that the two edges or the perimeter of that ellipse actually meets up in the middle to form a straight line, right? In a manner that's not very different from the way wounds heal, right? At this point, you can see the embryo twitching. This will now become a larva and hatch out of the shell. So what I'm going to tell you about today are two processes that we've been obsessed with. One is to ask, how does this structure here actually move from the head end of the embryo back to the tail end? So this tip here, which is closer to the left of the slide, actually ends up being at the right end of the embryo. The other process we're interested in is how do these two arcs of that make up the perimeter of the ellipse actually come and meet in the middle to form a nice seam. And what I will sort of illustrate is that morphogenesis relies on a fine balance between being deformable and maintaining shape. You can't be so deformable that you can't maintain shape. I'll also show you that it relies on an interplay between molecules and forces, and that it relies on an integration of information across multiple scales.
Can I ask a quick question, because this is connected to something that Sundar spoke sometime back in a colloquium. At this point also, there is like as the shape opens, as the thing opens up, unfolds, the symmetry of the entire thing is still maintained. So there are the symmetry of the entire.
Yeah. Is it symmetric?
Yes,
That
it
was
is still
your
maintained, right?
Yeah. So if you watch that movie a little carefully, I mean, I play it again, and if you watch this line here, right?
Yes.
It's a little bit off center like that, but it becomes a little more centered as time goes by. So what I'm trying to say is that there are asymmetries during this process that fix themselves through mechanisms that we've not addressed for that part of the process, but have addressed for something else. Yeah.
Gravity plays any role in the orientation?
Say again, you'll have to speak a little loudly. I'm a little hard.
Do you think gravity plays any role in the, like, I don't know, orienting these things while
Say
they're
again, gravity.
gravity, the direction of gravity, like
Does it
maybe,
affect?
yeah. For example, if you did the same thing on the International Space Station, would it have a different structure?
Good question. So, well, there's a less sophisticated version of the International Space Station that's called a centrifuge,
Okay.
in which you can
Sure.
spin organisms. And yes, so gravity does have some effects. And some of the simplest ideas of, so if you have an aggregate of cells that you suspend in a hanging drop, right? You have an aggregate of cells that's settled down at the bottom of a drop that's hanging from a surface. You can see that the bottom part, or the one that's in contact with the bottom of the drop, behaves a little bit differently from the top of the drop. Aggregates that are suspended, embryos and aggregates that are centrifuged also have show defects in development. And I don't completely know what the space station findings are, but it's fairly clear that microgravity and gravity do influence embryonic development. Sometimes they mislocalize proteins and RNAs that need to be localized in very specific positions in order for this pattern to emerge. Okay.
Okay. Thank you. Oh yeah, Sundar.
So I'll just quickly add on to what you said for that question. So it really depends on the scale and the force required for a particular process that is happening. So sometimes gravitational force is too little for the kind of forces that are required to deform tissues into a particular shape and so on. So it really depends. For example, in early chick embryos, gravitational forces actually are important in specifying the posterior part of the embryo. Like Maithreyi, finished off that, that comment. There are certain proteins which respond to certain gravitational forces, which actually get localized to a particular location of the embryo, and that determines the future posterior of the chick embryo, right? So, but in other cases, gravity doesn't play a role. So you can actually, if you can actually measure the forces involved in any of the processes that are happening within an embryo, then you can, of course, gravitational force, you know, so you can compare and contrast and see what is happening.
Okay.
In this example, is it clear how the head and tail directions are broken, symmetry is broken? In this example that you showed, is it known what breaks the head and tail symmetry? So one side forms a head.
What breaks the head and tail?
What breaks the head and tail symmetry? Okay, we have to sort of go back to the beginning a little bit. So in fact, I mean, in many organisms, okay, so that it's also a little bit of a chicken and egg question. I'll sort of say that in the beginning. So I suppose, I mean, you could say there are proteins and RNAs that are localized to, let's say, the head end and the tail end, if I think of the fly. And that system is actually set up it's already present in the egg even before that egg becomes fertilized. there's a set of localized determinants in the egg prior to fertilization. Sometimes fertilization by the sperm can be a symmetry breaking agent. And you could also say that cytoskeletal organization, small changes in the orientation of the cytoskeleton can create a bias in the amounts of proteins at the head and tail end. So I suppose the chicken and egg question is whether forces and or polarized transport generated by the cytoskeleton cause polarized distributions of head and tail determinants, or is it all genetic? I mean, so either way, transport has to be biased for these proteins to be at the head and tail end.
Okay.
Yeah.
Let's continue.
Okay. Okay, so what was I saying? So I was saying before this that we're interested in these processes that on the one hand cause the movement of the future tail end of the embryo away from the head end, and on the other cause the movement of the two lateral sides of the embryo towards each other so that they meet in the midline and form a seam. Now, both of these transformations are happening in what we call epithelia. And just to tell you what an epithelium is, it's a collection of polarized cells. When I say polarized, I have a top end and a bottom end. And it's a mechanically connected sheet. You can think of them as polygonal cylinders that are connected to each other like this, except that this, drawn by a physicist, and it's really a toy model in that all cells look similar and this sheet is flat, right? You can describe each cell within the sheet as having a height, as having an area at its top end, an area at its bottom end, and so on. And you can think of cells of different shapes that are just characterized by differences in the geometry of this basic configuration, except that sheets in real life are invariably curved, and cells can't actually pack themselves like this. And so if you actually look at how cells are packed on curved sheets, you'd see something like this. Cells make sometimes multiple have different neighbors at the top end, different neighbors in the middle, different neighbors at the bottom, and so on, right? But just these sheets, they're monolayers, and the complex transformation of these simple sheets are what we're interested in looking at. And one of the things that we've been intrigued by is not just how you bring the tail end close to the tail, or how you form a seam that creates a nice covering around the embryo, but also within these processes, and I'll describe them as we go along, are a number of other patterns, that allow us to see that not all cells within these sheets behave in the same way. So here, when I'll show you in this movie, that this cell in the center, and we're just looking at the cell from the top end, the cell in the center actually, over time, acquires a different shape from the cells around it, right? Which are also transformed by that cell. So when I play this movie, you'll see that the cell in the middle becomes smaller with time, and then disappears from the sheet. The cells around it are transformed in shape with their long axis perpendicular to the interface with that cell that disappeared. So this pattern here, which is dynamic, is something we call a rosette, because it looks like a flower with time. And what you will also see here in these black lines is that one element of the cytoskeleton actually aligns and polarizes in the neighboring cells as this happens. So what does this mean? It means that within the same sheet of genetically identical cells, there's this cell in the middle that's doing something different from the cells around it, which means that there's morphodynamic heterogeneity within that sheet. And this is in a state when all cells in the sheet are genetically identical. So the question that it poses is, where does that information for heterogeneity arise? In this example here, we look at that same elliptical tissue. I've just turned the embryo around, but using some ways of visualizing small fields of cells, you can actually watch this process by which the two flanks actually come and meet in the middle to form this seam. And what I'll tell you, if time permits at the end, is how the cells here that lead the flanks, cells here and cells here, are different from other cells that are part of that sheet. The third example is this, where cells don't look identically shaped. You can already see that. But if you watch this movie, you will see that in fact the cells are oscillating. They exhibit shape oscillations. They're expanding and contracting over time. And if you're perceptive, you'll notice at a particular point in this movie that this juggling reduces, and I'll point to it. So it's sort of starting now. You will see that the juggling reduces. And then once that juggling reduces, you'll actually see that the cells become small quite rapidly and help bring the same two edges close to each other. So there's heterogeneity in shape, but of a different kind. And it's also temporally regulated, meaning there's a phase when they're juggling around a lot, and a phase when those fluctuations or oscillations dampen. And the fourth one, which I'll spend a little time talking about, is the
question.
Yeah.
What's the scale of the cells?
What is the scale? Okay, so I should have put that here. So each individual cell here, I would say, is between 10 and 20 microns across. Here, with these cells are the same cells as these and these. So these cells are also 10 to 20 microns across at the beginning, but you will see that they become smaller as time goes by. Yeah. And so this process I'm going to talk to you about a little bit. But if you watch this movie where just the nuclei are labeled and not the cells, I'll focus on the region that's red and green. And so what we're interested in asking is, how do these motifs form? How does a rosette form? How do you form a seam? How do you generate oscillations, and how do you regulate that in time? How do you build a structure that I'll highlight later, the placode, and how do you build that sort of onion ring layered organization within the sheet of cells? And so the things we're interested in asking is, how do you build in heterogeneity? How do you ensure collectivity? How do you order cells within a sheet to enable these motifs? What purpose do these motifs serve? Do they allow cells to be packed in let's say, thermodynamically favorable ways? Do they allow the tissue to become deformed? Do they generate flows? Do they generate forces? And from a molecular perspective, we're interested in asking how you can modulate the link between the membrane and cytoskeleton to allow, for example, if I just take the example here, that the interfaces that this cell makes with its neighbors behaves differently from interfaces that its neighbor makes with other neighbors, right? So these are the sorts of things we're interested in. And so today I'm going to take you through work of some of the people in my lab. I talk about the work of Shudeepa and Charvi for most part. I'll briefly tell you what Padma, Lamya, Anupriya, Upal are doing. Okay, so I'm going to start with this process of germ band retraction, where the germ band, which is the structure here, moves in this direction from a starting point here. So, and then ends up at this place. And so the purpose of this movement, so many embryos in development are folded like this. And the purpose of germ band retraction is to actually straighten the axis so that my head and tail are at two opposite ends. Even though we said that this was specified genetically very early in development, it actually requires cell movements for it to stay that way at the end of development. And so many tissues, for example, the gut relies on this process to be positioned properly, as do the segments of the embryo. These segments are like vertebrae and ribs, right? If you didn't have this process, I would be bent like a hunchback. And so, so we set out by asking what actually happens during this process. And to cut a long story short, we can, just by visualizing this process, watch how this germ band, and we've just labeled a red part at the future tail end of the embryo. So you can see where it starts and where it ends up. And I think one thing you would have seen is that it started off, the region at the head end of the embryo started off as a heart-shaped structure that looks like this. And by the time it reaches the end, it'll be a heart-shaped structure that looks like that. So there's actually a shape reflection. So you can see that this point narrows now and the heart is actually shaped like that. Yeah.
Is this an animation or is it an actual video?
This is not an animation. This is,
Sorry.
you take an no, that's fine. I should probably have said that. You take an embryo that has proteins that say localized to the membranes that are then tagged to a fluorescent protein, so that if you just put the embryo, it's a bit like an engineered firefly. Let's say you put the embryo down the scope and excite the fluorescence of the protein. You can, depending on where that protein localizes, actually see what the cells are doing. And so one of the things you saw was that that two tissues, sorry, just a second. Yeah, you saw that this green tissue here almost lifts off from this other region like a veil, and it sort of straightens out, just as this part of the tissue straightens out or unfolds and disappears to the other side of the embryo. And this process here is important because when this fails, an embryo looks like a curled up porcupine, right? It's just permanently curled up like that. And the people who first mutated the genome identified genes in which this process is disrupted and gave them interesting names like hindsight, because the hind is inside, or U-shaped, or tail up, because the tail is on top rather than at the back. And this sort of unfolding or straightening is important, or is also seen, is a part of development in the fish. And in humans, if you don't straighten the axis, you can have very severe congenital kyphosis. It's also required to position the internal organs. So now I'm going to sort of try and move quickly to ask how does this germ band retract? How does the axis straighten? And the way we went about addressing that is to look at what the kinematics of this retraction is, how the tissue topology and geometry change, and to look at how the behaviors of cells within that tissue change, right? And this is when we saw that different cells behave differently, even within a small region. So one of the first things we did was to look at just this part of the tissue, but to use nuclear labeled movies of embryos in retraction to get a sense of how rapid or what the velocities of retraction are. And we did that by using particle imaging velocimetry. And you can see that you can work out what the velocities are, what the direction of this retraction is. And the thing that actually told us is that this process starts off at a very small velocity and then picks up pace to reach a maximum velocity and then actually slows down. And that told us that the speed of retraction is temporally regulated. And one of the things that we've buzzed by now is to ask, how does this tissue regulate its velocity? For example, in the same way that a vehicle can regulate its velocity. What keeps it slow for some part, what keeps it fast, and then what causes it to completely slow down. What also we saw was that if you label different cell fields within this, and here you see some stripes labeled, you see that the stripes actually change their shape and position over the course of this movement, right? And so I play this in a higher magnification, where here is the future tail end, and you can see how that heart-shaped structure here, which will keep coming back to the front because we shifted the frame. You'll see that this heart-shaped structure elongates and then becomes that inverted heart shape as it finally reaches the tail end. And so if you capture this in stills, you will see two things, that this heart-shaped structure changes its shape. That's the magenta color that you see here over time, and eventually moves away from what it was closest to, which is this elliptical tissue, right? What you'll also see, if you look closely, this heart-shaped tissue is that has a very interesting layered organization, where cells close to the middle here are more isotropic in shape, whereas cells away from the middle are very anisotropic, right? So you can almost see a layering, if you will, in the shapes of cells. What you'll also see over time is that cells that are close to the middle here actually end up being closer to the tail end later on, while cells that are farther away from the middle end up being closer to the head. So in a way, you're converting a sort of vertical information to horizontal information. And so if you spend some time looking at these cells more carefully, you'll see that one thing that distinguishes these cells is that the yellow cells that are on the periphery have larger aspect ratios compared to the cells in the middle that stay relatively isotropic. The middle cells are somewhere in between. What also distinguishes these three populations is the proportion of cells that look hexagonal. Why are we worried about hexagonal cells? The sided, yeah.
One thing, this is the density of the cells. The number of cells is also increasing as a function of time.
Say again?
The density of cells, the number of cells is also increasing as a function of time, right?
In that region?
Yeah, in the entire cell. You're having cell divisions, all of that is
Okay.
increasing. you have a velocity plot which goes up and comes down. Do you have an idea? How does the number changes? Because as the number changes, the cell is becoming more stiffer. So,
Okay, so I'll say a couple of things. In this process, there's only a narrow rim of tissue in which cell divisions are happening. So we don't think that the cell numbers are increasing by a lot. We do think that progressively, if you just look at this region, for example, the density of cells in the blue region relative to the gray region, relative to the yellow regions are a little bit different. And if you look at the number of neighbor cells have in these three regions, in the blue region, the cells are predominantly six-sided, whereas in the regions, in the gray and yellow regions, they deviate away from being hexagonal. The reason we're also looking at sidedness is because in a tissue where the packing is stable, you have predominantly six sides, but in a tissue where you have rearrangements happening, you deviate from six-sidedness and you have cells that are 5 or 7 sides. And so what that tells us is that potentially as you go away from the center, you have an increasing fluidity within that sheet, with the ability of cells to rearrange being used as a proxy for fluidity. And this was something that Mandar Inamdar at IIT Bombay actually allowed us to strengthen by showing that if you look at the pure shear magnitude along this axis here, right, going from this region to this region, there's a small but significant increase in the magnitude of pure shear as you hit this region here. So what this allowed us to say is that these cell shape gradients are likely also accompanied by gradients in fluidity within that zone. But the central region of the structure is relatively more rigid compared to the periphery. Okay, so
flattening is by pure shear. That's what's causing the area to go to zero.
Say again?
The flattening of your cells area, that the area
Yeah.
goes to zero, is by the pure shear mode.
is what?
Is by the pure shear mode of the system.
Is it by? I don't know about the by, but we know that it does happen through a process called T1 transition. So interfaces that are radially oriented shrink, and then stretch happens in the perpendicular direction. So whether that's generating the shear or whether shear from the regions around is causing that intercalation is something we don't yet know. Okay, so maybe I should move quickly because I have very little time left. So I think what we worked out was that you have these morphodynamic and mechanical gradients that exist in this region of the germ band that's actually translocating the farthest. And what we also saw was that in the other direction, which is that along the top to bottom axis of the cell, the heights of the cells are also different between the central, middle, and lateral, right? So that allowed us to say that you have this organization that creates curvature on two sides, one that's a little curvature here, and another that's a more extreme curvature here. And the structure maintains its coherence as it moves during retraction. And so we wanted to ask what keeps that structure intact, and is the intactness of that structure necessary for this process? And so if you look at actin or myosin around that structure as this process progresses, you will see rings rings or cables of actin that form around that structure, right? And that anisotropy of the cytoskeleton actually recapitulates the anisotropy of cell shape. So you have cables that appear to fence the central region from the middle region and the middle region from the lateral region. And that is the function of those cables is evident when you use genetic tools to reduce the formation of those cables. What you see then is, sorry, what you see is that when you, yeah, what you see is that instead of this nice radial organization of central middle lateral cells, when you perturb contractility, cells look all mixed up, right? You don't have a clear layered organization. And likewise, if you just cut one of these cables, you see that that part of it is not just twisted, but it also has a jagged edge. But the moment you build a cable back there, it starts to look rounded up and coherent again. What also happens when you mess up with contractility is that you increase the propensity of non-six-sided cells in all three populations, suggesting that maybe you're fluidizing this field of cells and therefore not allowing regions to maintain their coherence. But the surprising fact is that if you interfere with these cables or contractility, you actually speeden up the process. So the duration of a part of retraction is much lower compared to normal contractility, suggesting that these organizations must function as brakes. And I'm going to cut a long story short and ask how might these organizations serve as brakes? So typically, the region that we're looking at is what resembles a plateau. That's just a local thickening of the epithelium that acts as the starting point for a structure. But what we see is that during this retraction process, this also acts like a brake. And this is what a disc brake looks like. And this is what a plateau looks like in two different views of the same thing. And we know that brakes use friction to decelerate. So one of the things we're interested in asking is how might this structural organization actually act as a brake? And so in biological systems, forces, traction forces, as I told you already, are generated by a class of adhesion molecules called integrins through their interactions with extracellular matrix. And the traction between two surfaces depends not just on the composition of each surface, but also on other properties like texture, macro texture, micro texture. And so one of the things we've been interested in looking at is how...
Traction, is it a velocity dependent or is it displacement dependent?
Say again?
The traction, is it velocity dependent or is it displacement dependent? In dry friction, traction is displacement dependent.
Right.
In your system, is it displacement or velocity dependent?
Okay, so I'll say, so I think we've sort of not resolved this, but so if I look at this region here, so there's one part of the substrate which could sort of provide within quotes wet friction because there's fluid here. Another substrate here which would do, which would sort of mediate dry friction. we've not actually, I don't know the answer to your question. But what we're beginning to think is that, and when I play this movie, you will see how the tissues sometimes interact with the surface on top, but then fall off and interact with the surface at the bottom. So they're dynamically switching substrates till they sort of become completely free from the substrate on top and are only interacting with the substrate below. So the things we're really interested in asking is, where are these substrates located? Are they located in different amounts in different places? And the short answer to that is yes. We've regions where you have, if you think of green, if you just focus on the green and brown, the green and brown are distributed differentially here relative to here. And so we think there are regional differences in the enrichment of traction generating substrates. Whether they contribute to the generation of different traction forces or not remains to be determined. What we have tried to do, however, is to modulate these traction forces using genetic ways. And one of the ways in which we've done it allows us to see that you disrupt this organization of cells in the germ band. What you also see is that the gradients that I previously showed you become flattened when you disrupt these traction generating interactions. And so what we're really trying to ask, okay, and what it also does is appear to make this movement unsteady in the sense that the velocity reaches a point, reduces, picks up again. So rather than there being a pattern of up and down, there's some unsteadiness, which we think could indicate that there's some sort of slippage. But what we really want to know is whether, in fact, differential traction mediates these effects and what are the properties of these tractions. What properties of integrin ECM interactions contribute to these differential tractions, and importantly, how can we measure them? So I think I will stop here, but if I can take 2 minutes, I'll just tell you about a few other things that we're doing. So in this process of building a seam, we're sort of interested in asking how cells pack themselves up at the new seam. We showed earlier on that the way these cells make contacts once they meet in the middle is through an alteration in the morphology of those contacts from head-on contacts, which are T or plus shape that we think are unstable, to an interlocked configuration, which we think is more stable. But in 3 dimensions, we still don't know how these two cells, the sort of quadrilateral shaped cells here, actually come and meet in the middle all along their top to bottom axes. And Padma is actually sort of rendering the 3D shapes of these cells at the edge before, during, and after this fusion process to try and understand whether the formation of this seam relies on going from cells that are irregularly shaped prior to formation of the seam to becoming more regular after the fusion has happened. We've also been looking at how you generate pulsatility, and in particular, what governs how deformable the membrane is and what regulates the amplitude of these oscillations. And we recently showed how the interplay between the microtubule and the actin cytoskeleton governs the amplitude of these shape oscillations. With respect to delamination or this rosette formation, we've been interested in asking how you can make these interfaces of a cell different from other interfaces of the neighboring cells. And you can see that several properties of the cells of the interfaces of these cells are differentially regulated. So these interfaces shorten with time. These interfaces away from this don't change their length. And these interfaces here lengthen with time. So in a way, what we're interested in asking is not just how cells change shape, but how within a same cell you can generate interfaces that have different lengths. And we're looking at how signaling pathways and cytoskeletal organization contribute to this. And finally, not all types of delamination happen through this formation of nice symmetric rosettes, as you see here. And Anupriya is characterizing these in greater detail. But what Lamia has shown is that in delamination that accompanies the formation of the nervous system, you actually don't have these symmetric rosettes. And you lose neighbors as the cells delaminate. And so what we're interested in asking, along with physicists, is how does the force balance shift to create these asymmetric rosettes, and whether the morphodynamic state of a tissue is responsible for these differences. And I will stop there and thank a couple of other people. I'd like to thank Bobby, who's managed our imaging facilities to allow us to do all of this imaging. I'd also like to thank Zwedi, who's allowed us to look after our fly stocks. And most importantly, I'd like to thank TIFR for funding all of this work, but also allowing us to pursue the questions we want to pursue. And I'd like to thank all the people in the lab who did the work, and my present collaborators, Vijay Krishnamurthy at ICTS and Mandari Namdar.
Thank you very much. Thank you very much, Maithreyi. It's absolutely in-depth work, and there's so much. I think there should be more interactions with the community within TIFR. I think there is so much interest of the amount of data that you're generating in terms of the morphological dynamics of this growing embryo. I think there is a lot of physics in it. So I think there should be a lot more, because these are very deep phenomena, that heavy phenomena. So thank you very much for sharing some of them. Questions?
I ask.
Yes, and then I'll take.
Okay, so many things which are coming in mind, but one thing which I want to ask you is that, of course, this cell movements that you're talking about, something is growing, something is reducing, and so on. And all these cells are, of course, they are shared by common, I think, boundary.
Yeah.
So, and inside that, there is this plasma, which is not maybe uniformly distributed, or density variations.
You mean between the two cells?
No, within the cell, within each cell.
Huh.
So what I was wondering is that, I mean, when something grows or something moves, is there a transfer of material from one to the other? And what is that gene that is controlling? Is it that, like, I mean, when you have a certain kind of shape of a cell, of course, I mean, inside this each cell, of course, things are not distributed uniformly, I guess. So, I mean, the gene that it controls various aspects. So, I was just wondering what, I mean, if you have
Okay, so when you say transfer of material, are you wondering whether there's a
Otherwise,
change
how
in
something
volume,
shrinks.
for example?
Yeah, how something shrinks.
Okay, so for most of what I described, we do not think that any of these changes are accompanied by a change in volume, but they're accompanied by a sort of redistribution of the volume that is maintained by sort of creating, let's say, physical, I won't say barriers, but if you think of the actin cytoskeleton as something that stiffens a part of the membrane, then
Okay, maybe can I take it? Say one example you showed that there are two cells. It takes the shape of a flower.
Yeah.
The middle one vanished at some point.
Yeah.
So how did it vanish? I mean, I was just wondering.
So this is with respect to which cell? Sorry.
Delaminated.
Delaminated. How can you?
The central one.
Yeah.
The central one vanished. I mean, it's shrinked and at some point it disappeared.
Right.
So I was just wondering how.
Yeah. So actually, I've so far only shown you sort of view from the top. So if I were to cut, these cells are all three-dimensional entities that you could roughly model as polygonal cylinders. So what is actually happening is that initially the cell has roughly the same area as its neighbors. As these interfaces are constricting, the area of the cell in the middle is becoming smaller and smaller, while the cell is still connected to its neighbors through cell-cell junctions. As time goes by, you sort of loosen the contacts between this cell and its neighbors. But at the same time, the interfaces of its neighbors are sort of bulking up on the same adhesion molecule. The idea is that the process is coordinated in such a way that there's never a hole in the sheet. And just as the cell becomes small enough at one end, so it forms this teardrop morphology, the other edges are close enough to make contact. Because one of the things that a tissue does not like to do is to have a hole in it, because it can serve as a seed for a bigger hole. If you have a slight increase in some sort of stress.
That is correct, but the volume, you said it's volume conserving.
It's volume conserved. So, okay, so the point I was trying to make is that its area reduces at the top end, but the cell actually becomes like that when its neighbors are sort of like that. So it increases along this top to bottom or apicobasal direction.
Also the flow that you are showing, there must be certain kind of force acting on it. So how that force comes
to create the cell to delaminate. Okay, so that again is a very good question. We've sort of looked at this for a long time. So I'd say two things. We don't actually understand yet what makes that cell decide, or what causes that cell to exhibit this behavior. But what we have shown in other ways is that if you tweak with the interfacial tension of a single cell, you can sort of induce it. If you tweak either with adhesion or with contractility in a single cell, so you can then trigger it to go down. In the normal situation, whether these stresses, and also if you use lasers to break the membrane cytoskeleton links, you can cause that cell to exhibit this behavior. In the native state, whether the neighboring cells are actually sort of forcing the cell out is also a question we've considered. And we've shown that the neighboring cells at least provide some of the force for the cell to go down. Although the initial forces for the contractility are generated by the cell itself.
Yeah, so this may be sheer ignorance. Now that you have a large amount of data, is it possible to think of some kind of modeling and use that, sort of given the data from a model and then use the model to predict something a little bit different? And is it possible something like that? I mean,
so with the amount of data you've generated, is it possible to model the data, maybe this mechanical forces data, and then predict something which you have not done experimentally?
Yeah, no, that's a very good question. One of the things that we did try and do. So this type of event actually is, we can't predict which cell in that sheet will actually go down. And so one of the things that we did ask was whether geometry or number of neighbors might be important. And some of the things that we are trying to do with Mandar and Vijay is to sort of try and in a vertex model generate different boundary conditions to see if one or other condition increases the propensity of this type of event, and from there sort of go backwards and see if we can predict where those events will happen.
There has to be some exchange of material. Is it possible?
Exchange of material between
some genetic information
between the cell and its neighbors.
Yeah. Maybe.
No, between the cell and its neighbors, yes. Are you talking about communication or a mass transfer? If you're talking about communication, there's clearly communication of both the chemical and mechanical kind, and it's just enough to take an optical tweezer and pull on an interface to change what a cell does.
Yeah, so mine is very general question. We know structure formation generally controlled by physical forces. Now, in all this, you're talking about physical forces, but you're also bringing in genetic information. So where does genetics decide which way something should go or which way it should not go? If it's just a question of, forces between molecules and surface processes and so on and so forth, so where does the deciding authority of genetics, where does it come in?
So, so I'd say the first point I would make is, if you think of forces, for most part, we're thinking of forces that are generated intracellularly.
Exactly.
And the forces are generated through the action of molecules. So in other words, the forces, of course, I can go poke a cell. That's some external mechanical force. I can go poke a cell, I can stretch it and so on. But for most part, we're talking about forces that are generated inside the cell through the action of these polymers and their interactions with motor proteins. Obviously, these are proteins, they're made by genes, and different cells are programmed to make some or all of these proteins. So genetics, one sort of top-down control that genetics exerts is to say, okay, cell X has protein ABC that can generate this type of force, cell Y has something else. But in addition, but we also know that forces between cells can feed back onto genetics. So I guess the way we think about it is that genetics is necessary to build the force generating machinery, but the force generating machinery can, in addition to change shape, can also go back and influence what happens in the nucleus. Yeah. So the simplest experiment of that kind is if you take a particular type of stem cell and just grow it on agar of different percentages, right? Soft to stiff, you can change what becomes of those stem cells. No chemistry has been changed. Just the fact that the agar on which you're growing those cells is in one case low percentage, in another case higher percentage.
I will just like to ask the last question. The idea that the force generating machinery is also a sort of
being modulated. There's genetic control on that. However, there is also feedback that also talks about some molecules that are reporting the cell shape at all the time in real time to the parent cell, right? So there has to be that as well in the genetic machinery that should control.
Yeah, so I didn't sort of talk about that. So, but one of the slides I did say the same molecules, the adhesion molecules, don't just link to the actin cytoskeleton to generate force. They're also mechanosensors. They're very tuned to what's happening in their neighborhood, and the properties of those bonds can be modulated by this by these forces.
Right. Okay.
One real last.
Yeah.
So you should really have the last word.
No.
No, I'll tell you why.
Yeah.
So thank you very much. Are we closing the colloquium?
Yes, we are closing.
So I just want to say that all of us should wish JD a very happy birthday.
No, and I would like to congratulate Maithreyi and the team for generating such exciting data that we should be continue talking about it more. So thank you.